CellQualia Intelligent Cell Processing System is the world's first instrument for automated cell manufacturing with process analytical technologies. This promotional video will explain how the system works and delivers a high level of quality control. In the field of large-scale cell manufacturing, there remains many issues. For example, cells continuously grow regardless of the daily life of workers. Open cell handling needs a skilled operator to avoid microbial contamination. Scale-up culture is labor-intensive. Frequent change of culture environment will be caused by moving in and out of the incubator. Process control relies on personal experience, and risk of rejection at final QC due to lack of intermediate QC. CellQualia Intelligent Cell Processing System, composed of refrigerator and incubation modules, will provide solutions for such issues in cell manufacturing by automatization of cell culture from seeding to harvesting, closed system for aseptic operation, auto-passaging, real-time imaging, inline process analysis technologies, auto-sampling function for offline analysis, and this system can be connected to a higher-level manufacturing control system. Representative cell types suitable for this system are mesenchymal stem cells, which need to keep original characteristics for therapeutic effect, and ES-IPS cells, which need to keep pluripotent status as raw material for a variety of cell products. The system will also be applicable to other adherent cells. For better understanding of the benefits of this system, an important key word is quality by design. Quality by design is an approach to monitoring and optimizing the production processes, and it reinforces the necessity of building quality into a process and provides a framework to ascertain early signs of quality issues. Since stem cells often change their characteristics in response to a variety of stimuli, application of QBD is essential for their manufacturing. SINFONIA is collaborating with the Foundation for Biomedical Research and Innovation at Kobe, Dr. Shin Kawamata, for the development of the CellQualia Intelligent Cell Processing System. Sampling function for offline analysis is a unique feature of this system, and LC-MS analysis for kynurenine and 2-AAA is a good example. Kynurenine, a metabolite of tryptophan in the culture medium, is reported to be a good biomarker for the pluripotent state of ES-IPS cells. When cells start a differentiation process, the earliest differentiation is toward ectoderm. Kynurenine level will decrease by the activation of its catabolic pathway, and 2-aminoadipic acid will accumulate in the culture medium. Therefore, suppression of kynurenine production and accumulation of 2-aminoadipic acid can be QC markers for a human pluripotent stem cell expansion process. This was reported by Dr. Kawamata's team. For your better understanding, system overview will be shown here. Unit 1 and Unit 2 in incubation module are houses for cell culturing. Reagent bags, sampling and harvesting bags are set in refrigerator module for preservation. Media and reagent buffering bags and seeding and passaging bags are set in room temperature area. Drain bags are located in the bottom, and CO2 unit, camera, tablet PC for system control, and main switch and barcode reader are as shown. A fully closed system is the other important feature of this system, and the connection diagram is shown on the right. For the user's convenience in installation, a tubing set with reinforcing plate is divided into 10 pieces called templates and constructed appropriate setting of templates and flasks, and aseptic connecting lines makes a fully closed liquid transfer system. All the records, including raw materials, are stored in the preferred digital format, and this system can register raw materials by scanning barcode with reader as a preparation for cell manufacturing. Registered material data can also be available at the next run like a database. At cell manufacturing, culture recipe needs to be set and registered in advance. In list view, registered recipe can be found, and each recipe is composed of camera control, setting of seating volume, parameters in ECM coding, and so on. User can recall registered recipe and edit them. Timetable view is very helpful in confirming the timing of instrument control. Automated cell manufacturing follows the cell culture recipe, and its workflow will be shown. At first, a seeding bag containing pre-cultured cells is set for cell expansion in Unit 1 with imaging, medium analysis, and medium exchange. At an appropriate time, cells will be harvested and ready for the next step. The harvested cells will be transferred to Unit 2 as auto-passaging and used for cell expansion in Unit 2 with imaging, medium analysis, and medium exchange. At an appropriate time, cells will be harvested and ready for the next step, or returned to Unit 1 to expand the cells further with 36-layered flask as an optional step. Data of culture environment and cellular status will be collected and used for system control and culture monitoring. Electronic data can be used for process validation and product QC. These are very important for QBD to be established. This is a screenshot of the instrument status monitoring view. Flow of medium transfer is shown as a bright broken line. This is a screenshot of inline monitoring view. The result of time course measurement of pH, glucose, lactate, and pH-glucose ratio can be monitored in separate windows. This is a screenshot of a real-time imaging data view. Imaging positions at the bottom of vessel are shown as current and registered ones. Focus, contrast, and brightness can also be changed in addition to imaging position. A single campaign of cell manufacturing will generate numerous data for process monitoring and evaluation. Biomarkers are biochemical indexes of cellular status. Culture environment data represents instrument status throughout the campaign. And cell imaging is for healthiness and growth rate of the cells. All parameters measured relate to cell quality during cell manufacturing and can be used for setting design space. In general, quality of the cells manufactured needs to be confirmed by QCSA at the end. For example, MSCs manufactured need to be checked with the criteria for MSCs proposed by the International Society for Cell Therapy. Among 3 criteria, cells adhered and grew on plastic surface, and the cells showed high positive expression for positive markers of MSCs. These results showed that the cells manufactured with CellQualia Intelligent Cell Processing System kept their characteristics as MSCs. For iPS cells, we also have confirmed the quality of cells manufactured with our system by a typical morphology of human pluripotent stem cell colonies, growth rate, markers of human pluripotent stem cells, and secretion of kynurenine rather than 2-amino adipic acid in the media. Analysis of cell surface markers by flow cytometry is a standard and numerical assay to show cellular status, but difficult to apply during manufacturing. Hence, it would be preferable to identify a more convenient but reliable index of critical quality attributes. Process analytical technologies, a unique feature of the CellQualia Intelligent Cell Processing System, will benefit users. SINFONIA is currently setting the recommended protocol for MSCs and iPSCs. Expected yield of MSCs is up to 500 million cells and up to 600 million cells for iPSCs at this moment, only with commercially available materials. At the next stage of our product development, SINFONIA is planning to prepare and provide fully integrated workflow management software. This system is designed to be an excellent interface to a higher-level system for manufacturing execution and control, and to realize Pharma 4.0 cell manufacturing. In progress view for each batch, User can check status of instrument, data from inline sensors, images of cells, overview of the batch, materials used, culture condition, and instrument log of events. CellQualia Intelligent Cell Processing System is also suitable for parallel manufacturing. If user sets 5 instruments, for example, status of each instrument can be monitored in machine status view, and running machine number over time is for efficient use of instruments. To identify potential critical process parameters, comparison of run charts would be essential. For example, data of 2 independent runs can be overlaid. The final goal of this is to set critical quality attributes, which are an index for the quality assurance of cell products. Data from the instrument can also be used to identify and prevent recurrence of problems. In Alarm and Warning view, the user can see all of the alarms and warnings in a summary table, capture frequency of alarms using heat map analysis for process and batch number, and timing and sequence of all events in event timetable. All the data of finished products will be stored as a database which can be viewed in Inventory view. User can see current status of inventory as summary by product type and summary by each batch. Delivery control is the other important aspect of product quality control. In Delivery Tracking view, user can see summary of each product, list of serial numbers, products in delivery, and their destinations on a map. So, the CellQualia Intelligent Cell Processing System will enable and be the cornerstone of efficient production management. Not only by manufacturing data acquisition and analysis for quality by design, but also data of materials, logistics, and quality check data to be transferred to a manufacturing execution system or laboratory information management system. Furthermore, input of clinical data for each lot will contribute to improving production management to a higher overall level. As shown in this video, the CellQualia Intelligent Cell Processing System is an ideal instrument for cell manufacturing and delivering the concept of quality by design. The system can be applied from process development to manufacturing for higher quality cell products.